talk about cardiac output, which is essentially a measure of how much blood the heart pumps out per minute. So within each heart rate, we know the various cycles, right? We know that there's ventricular filling versus ventricular ejection. And so cardiac output measures how is the heart doing in terms of its job as a pump. How much blood is it really pumping out to the systemic circulation, systemic organs, and tissues every minute? Now, we know that the formula for cardiac output is stroke volume times heart rate. And what that means is how much blood the left ventricle specifically is pumping out with every heartbeat. And the reason we look specifically at the left ventricle is because this is the chamber that directly feeds the entire systemic circulation. Okay? And now when you look at an example of, let's say, stroke volume, we know that a typical stroke volume is about 70 milliliters. And we know that at rest, the heart rate is usually about 70 to 72 beats per minute. And so our typical cardiac output is about 5.5 liters by that calculation. Now, we also know that the entire blood volume is also about 5.5 liters. And so what does that tell us in terms of cardiac output versus our blood volume? What would that tell us? What can we deduce from those two facts? So the average cardiac output is about 5 liters, and then our average blood volume is about 5 liters. Perfect, right. So the entire blood volume, about 5.5 liters, flows through your heart. It's pumped through your left ventricle every minute. Right, so the entire cardiac output is just about the size of the entire blood volume, which is pretty amazing. Right? And so cardiac output is really a measure of how well the heart is performing as a pump. Now today we're going to really explore the factors that affect cardiac output, and we're going to talk about them independently. So we'll talk about factors that affect the heart rate first, and then factors that affect the stroke volume. But before we do that, we want to understand that the heart has two means of regulating itself. There's intrinsic control, and then there's extrinsic regulation or control. Now, extrinsic control is factors that affect the heart that are external to the heart itself, such as nerves. We already looked at the autonomic nervous system, so we know that autonomic nerves, which include parasympathetic and sympathetic nerves, innervate cardiac muscle, innervate the heart. And so they are a measure of, or a form of, extrinsic regulation of the heart. And then we also know that hormones such as norepinephrine and epinephrine play a part in that neuronal input as well, play a part in that autonomic regulation of the heart. So those are two factors that are external to the heart, right, not a part of the heart itself, that can affect contractility, that will affect heart rate, and how the heart behaves in total. When we look at intrinsic factors, this is what we call autoregulation. And this is this idea that the heart can regulate itself, regulate its own behaviors. And we'll talk more about a specific example, which is Starling's law. But basically, the heart can determine its own rate, its own rhythm, all intrinsically. And we'll talk about how that's brought about when we look at Starling's law later on. So those are the two forms of regulation that the heart exhibits. Intrinsic control, which is regulating itself, versus extrinsic control, which is basically nerves and hormones that have an input on heart rate and contractility. Now this schematic diagram should be a little bit familiar, and it's showing us how the autonomic nervous system influences the heart. Now the take-home point here, or the important points here, is that we know the vagus nerve, which is a direct flow from the parasympathetic division of the autonomic nervous system, it's going to innervate the SA node and the AV node only. Okay, whereas the sympathetic division, which we should be familiar with as T1 to L2, running through the sympathetic chain, that is going to innervate the SA node, the AV node, but then also the ventricular contractile cells. So this is something we studied before. We know that there are two types of cells that make up the heart. Some are pacemaker cells, typically found in the SA and AV node. Some are contractile cells, which are found actually throughout the myocardium. but the actual muscle of the heart wall. And so the message here is only the sympathetic nervous system innervates the contractile cells of the heart. The parasympathetic nervous system innervates only the SA node and the AV node, so it does not innervate the contractile cells of the heart. Okay. So we'll start by looking at factors that influence the heart rate, and then later on we'll talk about factors that influence the stroke volume. So heart rate is basically how many beats the heart makes per minute, how many times the ventricles contract per minute. And that is a reflection of the SA node. We know the SA node is the autorhythmic group of cells that sets the pace for the heart rate. And intrinsically, the SA nodal cells want to fire 100 times in every minute. And we say intrinsically because if you took a SA nodal cell and you put it in a petri dish, if it was not in the heart, it would fire an action potential every 100, or it would fire 100 action potentials every minute. In the body is a different story. So when the SA nodal cells are in the body, they are under the control of the autonomic nervous system. And you know that at rest, the parasympathetic division of that nervous system slightly dominates. And this is what keeps the heart rate reduced at about 75 beats per minute. So the alternatively, if the body is in an excited state, the sympathetic nervous system is slightly more dominant, and we have a higher heart rate. Okay. What we want to really understand here as well is that the heart rate can vary. It can vary based upon body position, body composition, your body state. If you're an athlete, you may have a lower heart rate, around 50 to 60 beats per minute at rest. If you're a baby or an elderly, you may have a higher heart rate. And the heart rate can go as high as about 180 beats per minute. So there's lots of variation and fluctuation depending on if you're at rest, if you're excited, and that has to do with the influence of the autonomic nervous system, right? The sympathetic versus parasympathetic. So let's look at an example of neuronal input on the heart rate. And this happens through the sympathetic activity. So if you increase the sympathetic activity, increase the outflow of norepinephrine or epinephrine, this is going to upregulate beta-1 receptors in the SA node, right? You know about beta-1 receptors already. These are adrenergic receptors. And beta-1 is specifically found in the heart, right? And so what these receptors will do is bind that norepinephrine. And the second messenger pathway here is to increase the opening of funny channels and increase the opening of calcium channels. Where did we see funny channels before? Where have we seen funny channels? Where have we seen funny channels and what are they responsible for? So you know funny channels allow the influx of sodium, and so it's going to help depolarize the cell. When we talk about the ionic basis of a pacemaker cell, right, we know that increasing the opening of funny channels allows more sodium into the cell, and that helps to depolarize the cell faster. So effectively, the sympathetic nervous system, using norepinephrine and epinephrine, increase the sodium and calcium channels, increase the positive ions moving into the cell, increase the rate of spontaneous depolarization, and that increases the heart rate. It makes it easier for these cells to fire action potentials more frequently, and that increases how fast the heart is beating. All right, let's look at a schematic representation of that, that second messenger pathway here. So again, this should all be familiar. The left side of this image should all be familiar to us at this point. We know beta-1 receptors are the adrenergic receptors that are found in the heart. We know that they bind norepinephrine and epinephrine, right? We know this is effectively the neuroeffector junction. So this is where a postganglionic neuron innervates cardiac muscle. And so what happens is norepinephrine binds to this receptor. We have a shape shift in this G protein. One of the subunits dissociates off, activates adenylyl cyclase, which is the amplifier enzyme in this case, And that is going to produce our second messenger, which is cyclic AMP. So we know this pathway already. Where we stop is what happens after protein kinase is made. What does protein kinase actually do? And so for the specific example of the heart, specifically for beta-1 receptors, protein kinase is going to activate funny channels as well as these T-type calcium channels, okay? T-type also means slow calcium channels, or fast calcium channels, I'm sorry. So T-type is fast calcium channels, L-type is slow calcium channels, and those terms are synonymous. So protein kinase activates these two receptors, and this makes it easy for the cell to depolarize, right? More funny channels means more sodium coming in. More fast calcium channels means that more calcium is coming into the cell. And we should remember that both funny channels and fast calcium channels are responsible for the action potential in a pacemaker cell, right? We talked about the ionic movement. And so essentially, making the cell easier to depolarize is what increases the heart rate. All right, let's talk about the opposite scenario, so the parasympathetic innervation here. Now we know the vagus nerve is the nerve that innervates the cardiac muscle. It's going to release acetylcholine, right? That's the neurotransmitter at the parasympathetic junction. And it's going to bind onto a muscarinic cholinergic receptor, right? This is the only type of cholinergic receptor at the neuroaffective junction. This opens up potassium channels, but then it also blocks or closes calcium channels. And that decreases the spontaneous depolarization of the cell. This makes it very hard for the cell to depolarize to fire an action potential. And so that decreases the heart rate. So the opposite of the scenario we had with the sympathetic nervous system is the scenario that we have with the parasympathetic system here. So first of all, the neurotransmitter is different. It's not epinephrine or norepinephrine. It's acetylcholine. It's going to bind to this muscarinic receptor. That's going to have a shape change in this G protein. And this is going to do two things. One is blocking the T-type or fast calcium channels. What do you think blocking those channels will do? What do you think blocking those channels would do in this scenario? So we know that opening up those channels makes depolarization happen easier. What do you think blocking that calcium channel would do? Slow it down. So it slows it down. It keeps the cell hyperpolarized. It makes it very hard for the cell to fire an action potential. Simply because we can't bring those positive cations in. We can't bring calcium in because that calcium is being affected. And so it's very hard to depolarize this cell. Right. Similarly, we also open up those potassium channels. What are potassium channels doing? What are potassium channels doing in terms of the ionic movement in this cell? Right, it's going to make it less positive or more negative. It's going to keep it hyperpolarized. And that's because it's letting all this potassium out. It's going to be impossible to fire an action potential. All of this positive cation is leaving the cell, and there's no positive ions moving into the cell. So it's very hard to depolarize, very hard to fire an action potential, and that essentially decreases the heart rate, making the heartbeat much slower. So this is how the parasympathetic system influences the heart rate. Now, if we compare those two autonomic divisions on the heart, and we look at this graph, we see three tracings. Now, the one in gray is showing how many times an action potential was fired at rest. And you can see that there are two action potentials fired at rest. Now, if we had only the parasympathetic influence on this cell, we would fire one action potential. Okay, you can see that in blue. Whereas if we had the sympathetic division influencing the cell only, we would fire three action potentials. And what does that tell us? That tells us that under the influence of the sympathetic division of the nervous system, which is shown here in green, we can speed up the heart rate by making it easier to fire an action potential. right? We can make it easier to fire an action potential, and so we can fire them easier and fire more in each second. On the other hand, if we're only having influence from the parasympathetic division, it's so hard to fire an action potential that in the same amount of time, we can only fire one. And that's why the heart rate is significantly reduced or decreased with the parasympathetic division. Okay, so that's heart rate. Let's finish up here. So the last factors that affect the heart rate here are hormonal control. And when we talk about hormones, we speak about epinephrine mainly. And epinephrine is going to behave the same way that it does as a neurotransmitter. The only thing here is that because it's found circulating in the blood, we call it a hormone, whereas if it's found at the neuroaffective junction or it's found at the synapse, we call it a neurotransmitter. But it has the same effect as we saw on this slide. Okay? So think about the effect of the neurotransmitter here being the same effect as the hormone epinephrine. It's going to increase the action potential frequency. It's going to increase the velocity of the action potential in muscle fibers, and it basically is going to speed up the heart rate, speed up the contractility. The other hormone that plays a role on heart rate is glucagon. Glucagon is a metabolic hormone. It's going to increase how cells uptake glucose. It's going to increase the heart rate. We're not going to get too much into the mechanism of how glucagon acts, but we just want to know that it has an effect of increasing the heart rate. This slide is basically pointing out the integration of both these divisions of the nervous system. This should be a review for us at this point. We know that the parasympathetic system influences the heart rate, so does the sympathetic system. And so at rest, our heart rate is a reflection of both these divisions, slightly more the parasympathetic side, so we know this.